So you look up this morning. Oh, that's not true.
So we are going to finish up our discussion of enzymes today to begin class, and then we're going to move into metabolism. So when we left off last time, we were talking about a specialized type of protein called an enzyme. Specifically, we were talking about how enzymes catalyze reactions. So how they speed up reactions, how they make them. So we were looking at the basics of what's going on when we have an enzyme versus what we call. So when we have a reaction, we start with our reactants. We want to turn them into products. Okay. Our reactants and our products have a certain amount of energy in them. And in order to get from the reactants to products, there's a certain amount of energy that they need to kind of like collect in the reactants to make them unstable before they will actually turn into the products. Okay. But without an enzyme, this can be an awful lot of energy. But with an enzyme, acts as a catalyst. We have a much lower activation energy barrier. So this reaction is going to happen more often, more frequently. Now we're going to look at the shapes of the enzyme and how that really works. Okay. So the simple model that people like to use, which I do think, despite the fact that it's not exactly right, is really helpful for understanding an enzyme, is that enzymes work kind of like a lock and a key. You have a spot in an enzyme that is the same shape as the substrate that's going to bind into it, and that's how they fit together. So this doesn't fully explain the complicated action of all of our enzymatic reactions, but I am going to just make sure that we can see what's going on. Okay, so our big old enzyme, the protein folded around, okay? And an important location on this enzyme is the spot where our substrate, our reactant for our chemical reaction, is going to bind in. So this is called our active site. So our active site is basically the pocket where the substrate is going to fit in. So in the lock and key model, that active site, that pocket, and the substrate are the exact same shape. Okay. In the induced fit model, there's a little bit of wiggle room, essentially. What the induced fit model is really a slight addition or a slight alteration of this idea. And all that is that it says that that active site and the substrate, when they come close to each other, interact a little bit so that that active site changes shape a little bit, kind of like Play-Doh, to match the substrate. And this is necessary in order to understand why some enzymes can catalyze reactions in multiple directions. So they can help you make the products, but they can also help you go from the products back to the reactants. And that wouldn't fully make sense with the lock and key model. But in either situation, you're going to have an enzyme, you're going to have an active site where the substrate binds in, and that binding is part of what helps us lower the activation energy and catalyze the reaction. I'm going to look at this picture to kind of show you, or try to show you, why this is helping to catalyze the reaction. So here we have a substrate and an active site that are slightly different in shape. We're looking at an induced fit model explanation, so that's okay. The active site just changes shape a little to match that substrate. Okay. But the catalyzing part happens kind of here. So basically, the idea is that by holding that substrate in place, that kind of fixes it so that when one of the other reactants essentially comes up to that enzyme as well, we're holding things in fixed places. And we can either stretch bonds or kind of push them closer together to make it easier for those bonds to form. So this is where the reaction would happen. Our product is a slightly different shape than our original reactant, than our substrate. So it pops out of that active site and our product is free here and our active site goes back to that original site. That's the basics of what an enzyme is doing. So now we're going to think a little bit more about how we can change the function of the enzyme. So we can change the function of enzymes and like help them along often using cofactors and coenzymes. So cofactor is a part of an enzyme that's made of something other than a protein. Usually this is like some kind of trace chemical that's going to be necessary for that enzyme to work. So basically what we're saying here when we have a cofactor is that the enzyme has a certain shape, but without this cofactor, the shape isn't quite right to have the normal enzymatic function that this enzyme performs in the body. So we need to eat like different dietary minerals in order to make sure that those enzymes keep working. Coenzymes are kind of similar. They don't have any catalytic activity themselves, but they're also something that's necessary for proper enzyme function. And they're going to participate a little more directly in enzyme reactions than our cofactors are. But I would say that the main difference when you're trying to figure out whether you're thinking about a cofactor or an enzyme is a cofactor. The factors really are going to be like simple metals and ions and stuff. Coenzymes, we're thinking things like vitamins. We're thinking more complex kind of organic molecules when we're thinking coenzymes. They're actually going to often transfer small chemical groups around. We'll talk about those as we get to them. But since you have all hopefully had metabolism before, you might remember acetyl-CoA, coenzyme A, right, is an example here of a coenzyme. An enzyme's reaction rate, we're talking about basically how fast it works, right? So we're thinking about how can we have our reactions happen faster? Why might they happen slower? There are a number of different factors that are going to kind of affect how efficiently our metabolic processes are going. So the first piece, the enzyme's catalytic rate, is just how fast does that individual molecule work, essentially. So like how many times per second can you have this enzyme take our little sphere and turn it into a triangle, right? So that would be the enzyme's catalytic rate. So number of molecules generated per unit of time. Now we can change the overall rate of the reaction by changing some things about the enzyme and changing some things about the environment that that enzyme is acting in. So remembering, our substrate is our reactant, right? Our substrate. So one thing that can make a difference when we're thinking about the rates of our reaction is how much of our reactant is sitting around. Basically, if we start with lots and lots of substrate, that reaction is going to happen quickly. If you only have 2 or 3 molecules kind of here and there for your substrates, your reactants, it's going to happen more slowly. Okay? So if you have more substrate to begin with, this is going to happen more quickly. Our enzyme concentration, we have the same sort of situation. If you only have 1 or 2 little molecules of enzyme floating around catalyzing your reaction, that's going to be a pretty slow process. If you have a lot more of those enzyme molecules floating around, they can grab lots more of that substrate. That's going to speed up a reaction. The third one, the affinity of the enzyme for the substrate, referring to basically the fit between the enzyme and its substrate. So how well they fit together. So affinity is like, are they friends? Do they like each other, right? So it might be the case that both our circle shape and our triangle shape happen to both be possible to fit into that active site. But the enzyme prefers to have a circle in there. So it's going to pick that circle over the triangle whenever it can, right? So that would say, we would phrase that as saying it has a higher affinity for that circle substrate than for the triangle one, okay? As we move forward and think about affinity in future lessons, we'll talk about it a lot when we talk about how hemoglobin moves around the gases in your blood. We'll find that there are a lot of sort of domino effects of an enzyme binding really well to a substrate or binding against it. But for now, we just want to remember that affinity of an enzyme, if we have high affinity, it means they bind together really well. So temperature and pH, if they go either too high or too low, will affect the rate of our reaction. Reactions in our bodies are pretty fine-tuned to operate at our normal temperature and normal pH. So really it's if we go outside of the range of our homeostatically regulated temperature or pH, that's going to impact our enzyme reaction. We are going to draw out a couple of things. So specifically about that affinity piece, I want to give us a couple of examples. But we'll talk about that substrate concentration, enzyme concentration. Okay. So when a chemical reaction is happening, you have to have your multiple reactants through different substrates bump into each other for a bond to form. A basic type of reaction, right? So if you don't have a lot of substrate, your reaction rate is going to be low because you're going to have to wait around until that reactant finds the other reactant. Okay. As you have more substrate, your reaction rate picks up. So this line is going up, right? So more substrate means a higher reaction rate. So this curve with a given enzyme, and we're starting with a low enzyme substrate affinity. We're gonna have a line, let's say like this, okay? So as the substrate concentration increases, the reaction rate increases. So if you have more of your reactant, you end up with more of your product. This reaction is going faster for a while, but eventually you hit a maximum rate, okay? So that's what we have here. It's the maximum rate of this reaction for this substrate with a low enzyme substrate affinity. Now we're going to compare it to what we see if we have an enzyme where we have a good enzyme substrate affinity, a high enzyme substrate affinity. So they really like each other, they really want to bind together. We're still going to start out at the bottom, right? We're still going to have a situation where if we don't have much substrate, we're not going to have much of a reaction, right? We have that same problem of if we don't have reactants, the reaction is not going to happen. So we start in the same place. And we're still going to have a line that's going up because as our substrate concentration increases, so does our reaction rate. But overall, this curve is going to sit higher, right? So as our substrate is increasing in concentration, so as we have more of it, that reaction is going faster and faster than if we have an enzyme that doesn't bind on very well to the substrate. Basically, because it can be more efficient, more effective. You don't have to kind of wait for them to want to jiggle into place. They act more like magnets. They like each other better. And you're also going to end up often with a higher plateau, right? So a higher maximum, right? Because that enzyme is just essentially more effective. So that's the type of curve we might be looking at when we're looking at things with higher and lower affinity to each other. Higher affinity, improving our reaction rate and increasing our chance. Those are things about the enzyme and the general physical environment it's in and that substrate that affect how the enzyme works. But we can also basically put our hands in the system and kind of meddle with it, right? We can regulate this even more by adding little pieces onto the enzyme, taking things away. Okay? So when we're trying to regulate enzyme activity within your body using multiple different cellular processes, there are a couple of different things we're going to consider. Now they are numbered 1 to 4, but I tend to think about them in kind of 2 groups. So I kind of think of allosteric regulation and covalent regulation as 2 contrasting things. And I think about feedback inhibition and feed forward activation as a separate sort of 2 group thing. But we'll run through all 4. Okay. So we're going to start with allosteric regulation. So in allosteric regulation, we have our enzyme. Okay, our enzyme. We have our active site that we've been talking about. So that's where our substrate would go, right here in this active site pocket. But what we also have is a separate location somewhere else on the enzyme that acts as a regulatory site. So at that regulatory site, that's a spot where some type of modulator, so some type of other molecule, when it binds in to the regulatory site, that affects a change on the shape of the enzyme, which is going to change how it acts with respect to its substrate, with respect to the reactions. We back up a little in our minds and think about the fact that enzymes are proteins that can maybe make this make a bit more sense. So the shape of the protein we talked about last time has to do with the fact that we have a certain sequence of amino acids. They start to fold first based on hydrogen bonding, but then based on different chemical properties of their R groups. So they're groups that are substituted that are different between our 20 different amino acids. So that's how we start to get a complicated 3-dimensional shape. Okay. So if you have some type of regulatory molecule, some type of other molecule that's going to get up to the enzyme and touch it from behind, right? It is also going to have some type of chemical property, right? Which in this scenario, that chemical property of the modulator interacts with the chemical property of the enzyme at the regulatory site and causes a domino effect where the whole enzyme changes shape a little bit. So despite the fact that the modulator is somewhere else on the enzyme and not at the active site, because that whole ball folded up together is all connected, because we started with that chain, when the modulator binds in, we can have a different shape of our active site. So it may not fit our substrate very well anymore with this. So allosteric, also, I think allo is like other, steric usually refers to like a place. So the name, if you know, I think these are Greek roots, but the name tries to tell you what's happening here. So regulation from another place on the enzyme rather than the active site. So that's allosteric regulation. Covalent regulation is slightly different in that in covalent regulation, rather than having some other molecule that just kind of sits into that regulatory site, we're going to have a reaction happen between the enzyme and another little group that covalently binds on the covalent regulator onto our enzyme. So it's going to permanently change the action of that enzyme until it gets killed off. In our allosteric regulation, we have that modulator go into the regulatory site, but we didn't form any covalent bonds there. So it might just pop out a little while later. So it can alter our reaction sort of temporarily, but it's not going to have necessarily a hugely long-term effect unless we have lots and lots and lots of that regulator molecule. But in covalent regulation, because we're adding on a chemical group by a covalent bond, we're going to see changed effects to that enzyme until that bond is broken. So what we see happening here is we're starting out with a chemical group and our enzyme. Okay. Then we have a separate enzyme that binds on that chemical group onto our original enzyme. So we can now see that we have chemical group, enzyme, and like in the allosteric regulation, our enzyme has altered activity because that addition of a covalent bond has changed its overall shape. And until we have enzyme B, another enzyme, come in and break that bond, get us back to the original, we're going to have altered activity. Basically, these are two ways we can change enzyme activity. In both of them, we change the shape of the active site, which is where the enzyme binds in to its substrate. Okay. In allosteric regulation, we have a modulator go into a regulatory site, probably pop back out of the regulatory site. In covalent regulation, we're forming a firm covalent bond to the enzyme. And so that change will be permanent until that bond is broken. Not going to ask you to memorize these words that are in red here, but I am going to tell you that usually when we have this covalent regulation happening, it's not just some random chemical group that's getting covalently bonded on. It's usually phosphates. Okay. So in future courses, you may talk about these at length. So an example of covalent regulation would be if we have an enzyme that gets a phosphate tacked on. The type of secondary enzyme that would do that tacking on, so that's catalyzing that reaction, is called a protein kinase. So the protein kinase would be forming the covalent bond. That's not a minus sign, that's a covalent bond here. That's forming our enzyme phosphate kind of complex. We have that enzyme phosphate complex until we have another enzyme catalyze the reverse reaction. So we're starting with our enzyme bound to our phosphate, and we have a phosphatase act, and it breaks that bond. So we're back to having our enzyme and our phosphate separate from each other. Another thing we could note on here that hopefully we remember from our previous courses. I'm not going to ask you to remember protein kinases and phosphatases, but you should be able to tell that that ACE ending tells you that this is an enzyme. So that's a way we name enzymes, is we put an ACE ending on it, telling us that it's working on that thing. So our phosphatase breaks off our phosphate, for example, is how that one got its name. Questions about these two processes before we move on to our feedback inhibition and forward activation? They're very similar processes. Slight difference, really the difference is the covalent bond. Okay. So now we're going to talk about feedback inhibition. And I think about these two things, the allosteric regulation and the covalent regulation, and then feedback inhibition, forward activation into separate groups, because the first two are talking about kind of mechanisms, like what binds in where, why did we change the activity? And when we're talking about this feedback or feed forward, we're talking a bit more about like the pathway of the reaction rather than shapes of molecules themselves. Okay. So what we're looking at here, again, we're starting with a reaction that turns our reactants A into these intermediate products B. They're catalyzed by enzyme 1. That's what this is here. Then we have a second reaction where we take B and turn it into C. And it has enzyme 2, catalyze that reaction. And then finally, our reactant C get turned into our final products D for this total pathway. So we will often not be talking about all the intermediate steps in all of the reactions we talk about in this class. They do exist. So if we were looking at this whole thing, we might say A is being converted into D as our product. When we're thinking about enzymes and how they work and how they kind of stabilize the speed of things, knowing that these intermediate steps exist can be important. So in this example, it's saying that the rates of E1 and E3 are constant. So nothing is changing about this first reaction and nothing is changing about this third reaction. Okay, so those are staying the same. Feedback inhibition means that something we're making in this process is going back and inhibiting or preventing some earlier part of the process from working as efficiently, as quickly. So what we see happening here is that in this example, B to C is the rate limiting step. B to C is the step that's going to be variable in rate. This enzyme 2 is the one that we can either speed up how well it acts or we can slow down how it acts. So in this example, what we're saying is that we make A into B, we make B into C. Once we have C floating around in our cell, some of it gets turned into D. But if C goes back and touches enzyme 2 again, it slows this down. So it would slow down B getting converted into C. So overall, we'd kind of be slowing down, turning C into D as well. So conditions we might want to do this in are basically this whole pathway in your cell is probably happening at a steady rate where everything is balanced well. Everything you got of A turns into B, everything of B turns into C, C turns into D. But if all of a sudden you end up with a lot of A, and therefore you end up with a lot of B, and therefore you end up with a lot of C, it could kind of like overwhelm this last step, depending on that enzyme. So you might want to slow down earlier parts of this process to get a more sort of steady pathway. Honestly, I like this example better. This is still talking about feedback inhibition, okay? But you can see, really, I think a little more intuitively how this would be helpful. So like, say this is a whole process, making ATP or something, right? So we have lots and lots of steps. In this case, 3 steps still. We have rates that are constantly going on. What this is telling us is that once we have made enough of this product, maybe I should have picked ATP, but this does happen sometimes with ATP. Once we make enough of this product, our final product can go back and inhibit the first step. So it can kind of turn off this whole pathway once we have made enough of whatever it is we're trying to make. So I do want you to be careful to not confuse the words feedback inhibition and negative feedback, but they are very similar and kind of related concepts. So we're thinking about negative feedback when we're thinking about regulating things overall, right? But negative feedback is we're seeing a change, and then we're going back to a set point when we're talking about homeostasis. Feedback inhibition is kind of like that, but for enzymes and chemical reactions, right? So feedback inhibition is a phrase we use specific to how these metabolic pathways are working. But we have the same sort of situation where some product we're creating is going back and kind of turning off earlier parts of the process. And that's more the level we want to understand this about. I'm not going to drill you intensely on long, complicated reactions. We really just want to know that we can have end products or intermediate products that go back and affect enzymes earlier in the pathway, and that the overall goal is really to keep things running smoothly, to control these reactions, to make the amount of whatever it is in the amount we want, at the speed we want, and to not end up with our reactions going in like weird little hops, okay? Because that just doesn't work very well in your body. So the final bit here is that there is an opposite of feedback inhibition. It's called feed-forward activation. It's rare. We're not really going to talk about it, so it's really here mostly to be complete. But what this is showing us is just that it is also possible that you can have a molecule created earlier in the pathway that jumps forward and affects an enzyme later on and makes it act more effectively. So these are both ways we could regulate the rates of different parts of chemical reactions in the body. When we're regulating rates in the body, we are regulating the action of enzymes. So before we move forward into metabolism, questions about enzymes in general, the sort of basic function of enzymes. As you think of them, let me know. But we are going to move forward and do some metabolism. Now, you should have had metabolism before, either in your biology courses or chemistry courses, or depending on what you took. So I want you to take 2 minutes to write down what you can remember about the main steps of cellular respiration. So a reminder that there are a bunch of steps, they have names. Cellular respiration is also known as glucose metabolism or glucose oxidation. What we're doing is producing ATP. So go ahead, think, look around, review a little bit, and try and remember which parts of this process produce ATP.
I don't know what I meant to hire. I always like bonus for studies. She has to get like. I more like. I'm going to the extra credit.
Three to nine during the week, is it?
Well, I know that.
So there's just 5 to 7, but the things are all posted up on.
But it's not gonna go.
Exactly, yeah, I know.
So the thing that can go in is.
Okay, so technically that's been a couple minutes, so hopefully you've had time to at least sort of evaluate your mental memory of metabolism and how much of this you remember. Hopefully you were able to remember some of the steps. Hopefully you remembered some of how ATP is produced. But if you could not remember any of this stuff and you could not remember where ATP is produced, you know what you need to look over. Correct. So before we get into our overall topic, I want to kind of center us. Okay. So as we're walking through, there'll be some numbers on here, but what we want to focus on understanding is like what cellular components, what parts of the cell we're in for our different steps. We want to really focus on which steps produce energy, produce ATP, and how much, like a lot, little, things like that. We're also going to be thinking about our electron carriers and why they exist, how they work on a kind of overview sort of level. So there are tons and tons and tons of enzymes involved in cellular metabolism. There are tons and tons and tons of little intermediate steps in each of our big sort of chunks of our cellular respiration process. We're not going to go super deep into all the details, but we want to make sure that we understand overall what's happening, why, what does it mean for the cell, and can start to predict, like, okay, what if a cell doesn't have a mitochondria? Can it still make energy? Things like that. Okay, what if your cell is in an environment where it really doesn't have oxygen because you're using all your oxygen because you're on a run? That's the kind of thing we're going to want to be able to understand later on after we've talked through and reviewed the basics. So we're going to be thinking about the role of ATP in our energy metabolism. We're going to be thinking about some of the main steps. And then we will add on how we can get energy from things that are not glucose. So we'll talk about metabolism of some other compounds. But we're going to start by thinking about ATP. So cellular respiration, also known as glucose oxidation. This is our aerobic cellular respiration involving oxygen, aerobic here. So our total big stereotypical reaction, how we usually get energy, how we prefer to get energy for ourselves, is that we take some glucose, our carbohydrate glucose. See our carbohydrate glucose, C6H12O6. We're going to use oxygen. 6 molecules of oxygen, and we're going to turn it into a bunch of energy, plus our other products, our waste products, carbon dioxide, and some water. Okay. So we're turning glucose into a lot of energy. We're making byproducts, carbon dioxide and water, we need to put oxygen. Energy that is usable by the cell at the end of this process comes in the form of ATP. So we're breaking down glucose, breaking the bonds in that glucose, taking the energy out and trying to turn them into ATP molecules. Okay. So we're going to be using the reactions in the course of breaking down that glucose molecule to synthesize, to create ATP adenosine triphosphate. I think we looked at a short picture of ATP when we were thinking about nucleic acids. But what we want to focus on when we're looking at the structure here, right, is our ATP molecule. We have our adenosine nucleotide. And then we have our 3 phosphate groups, so ATP. 1, 2, 3, okay? So what we are going to be doing at the end and throughout metabolism is we're going to be trying to tack on phosphate groups so that we can hold energy in this bond, because our cellular processes are often fueled by breaking that bond and using the energy to do something else. That's how ATP works. I am not going to ask you to do that chemistry math, but I do want you to realize that this formation of ATP, right, so making ATP is an endergonic process. So you need to put energy in if you want to make some ATP, which makes sense, right? Because what we start with to make ATP is our ADP. So we have our phosphate group, our sugar, and that was our nitrogenous group, sorry, our sugar, and then I'm going to put the phosphate groups here, 1, 2, 3, right? So this takes energy, you have to put energy in, it's endergonic, because you need to form that 3rd bond if you're starting with ADP, which had only 2 phosphate groups. This is also a condensation reaction because it produces water as its byproduct, which is part of why we saw water in that overall equation for glucose metabolism. We may also call it a phosphorylation reaction because what we're doing is tacking on that phosphate group, phosphorylation, adding that phosphate. And this can happen 2 different ways. And it does happen both ways within the course of our cellular respiration, our aerobic respiration. So the first way that we can form that bond between the 2nd and 3rd phosphate groups in ATP, something called substrate level phosphorylation. Okay, so in substrate level phosphorylation, what we're going to see is we're going to see ADP, our adenosine diphosphate. So that's our starter molecule that we want to tack on another phosphate to. So that's what we're trying to phosphorylate. And we are going to see a phosphate group that was bound onto something else covalently, just transferred on to our adenosine diphosphate, leaving us with some molecule that now doesn't have a phosphate on it, plus ATP. Okay. And we are going to see enzymes involved with this process, changing those bonds around, essentially. The special thing that we can do in aerobic respiration that makes actually almost all, or at least most of our ATP a much more efficient process is oxidative phosphorylation. So instead of moving a phosphate group from one molecule to another using an enzyme, we are going to use the electron transport chain. So a series of enzymes embedded in a membrane in the mitochondria that are going to help us build big proton gradient across that membrane, which when we release it and allow it to flow through a final enzyme, makes lots and lots of ATP. So this process we'll get to at the end of metabolism, gives us a lot of bang for our buck, but it's going to involve a bunch of sort of free floating phosphate ions. We still have our starter molecule ADP. just like we did on the substrate level. We're gonna end up with a bunch of ATP and no like molecule X left over because these ions are just kind of free floating. We'll talk about them as they're relevant to our different steps of metabolism. So ATP breakdown gives us some temporary energy storage. So when we break down ATP in a hydrolysis reaction, so when we add water to the ATP, we break off that third phosphate group. That leaves us with ADP, which was our original kind of starter molecule on the previous slide. It'll have that phosphate group broken off, and we'll get some energy out of that bond that we just broke. And that energy is energy that we could use to say, contract the muscle or start to contract the muscle. I'm not going to ask you to memorize this whole big equation, not the purpose of this course, but realize that we do make kind of on the order of 38-ish ATP molecules at the end per molecule, theoretically. So another thing to notice, so we are trying to turn ADP and phosphates into ATP so that we have this ATP to use elsewhere in our cells. We do lose some energy as heat in this process. So we're not perfect at capturing all the energy from our original glucose molecule. I am not going to ask you to memorize a ton of chemistry equations, so I'm going to just highlight what's going on here. So basically, the most important part for you to understand here is this bit about efficiency. So this process is not totally efficient. So body heat is partially just from this process of breaking down glucose and making ATP. Okay. So efficiency actually varies, like how much of that energy from the glucose you manage to trap in ATP. It's going to vary in different tissue types in your own body. It's going to vary in people of different ages. Might also vary just depending on your personal genetics. So there's some variation there. I want you to know that we are losing a fair amount of this energy. So there's some wiggle room. So now we are going to talk about the process itself. Okay. So our big stages that we're going to focus on are glycolysis. Then we'll have a sort of linking step in between glycolysis and the Krebs cycle. So we're going to convert pyruvate and bring it into our mitochondria. We're going to have the Krebs cycle or the TCA cycle. You might have learned about it using different terms before. And after that Krebs cycle or TCA cycle, we're going to run the electron transport chain across that inner membrane of the mitochondrium. That's going to do a lot of oxidative phosphorylation for us, which is where we're going to get most of our energy from the glucose molecule and turn it into ATP. So depending on the source you're looking at or the textbook, some of these terms are referred to in multiple different ways or multiple different numbers of stages. That's because it's a whole big long process and someone has to decide what deserves a name and what doesn't. But I'm not changing the process overall from whatever you learned in previous courses. What we have highlighted here in these big bullet points really are just the 3 steps that do produce some ATP. So that's why these 3 are here specifically. So in the first 2 parts here, in glycolysis and in the Krebs cycle, we're going to produce a little bit of ATP, not much. When we produce ATP during glycolysis or the Krebs cycle, TCA cycle, which is the same thing as the Krebs cycle, we're doing that by substrate level phosphorylation. So that's going to involve enzymes moving phosphate groups around. The electron transport chain, where we see our oxidative phosphorylation, so where we're going to be releasing a big proton gradient to make lots of big. We're in time, so we will start next time with glycolysis. So I do recommend, if you don't feel super comfortable about your memory of metabolism, go for that.